To understand what limits our cognitive ceiling, we have to map the exact psychophysiological substrates that govern working memory and fluid intelligence. We measure these baseline capacities using speed-based tasks. These require a subject to actively monitor incoming information and execute precise reactions while bombarded by constant interference and distraction. High fluid intelligence is often associated with a hyperactive neural response, the idea that solving complex problems requires the brain to run at maximum output. This graph charts cognitive load against the brain's localized blood oxygenation, or BOLD response. When we look at individuals with high cognitive capacity, we see a clear inverse correlation. Their activation curve is tightly constrained. These negative values represent task-induced deactivation. The brain actively suppresses irrelevant networks to manage cognitive demands. This lower overall brain activation is called neural efficiency, and it serves as a consistent neural characteristic of intelligence. Working memory and fluid intelligence are strictly bottlenecked by the physical mechanisms of attention regulation. Two primary systems govern this regulation: the locus coeruleus norepinephrine system and the balance of dopaminergic receptors in the frontal cortex. Together, these systems orchestrate how large-scale brain networks switch on and off. When they fail to communicate, the working memory representation dissolves. High cognitive capacity relies on precise neurochemical regulation that actively prevents interference, rather than raw metabolic horsepower. The locus coeruleus norepinephrine, or LCNE system, operates across all levels of brain function, from metabolic processes to the organization of large-scale neural networks. We can measure its baseline activity clinically by observing resting pupil size. The data shows that the general factor of executive attention is an accurate predictor of this resting pupil size. Executive attention is divided into maintenance and disengagement. Specifically, the capacity to disengage attention uniquely predicts the resting activity of the locus coeruleus. In individuals with low working memory, a physiological cascade begins with LCNE dysregulation, producing a weak signal when attempting to engage the frontoparietal control network. Because this frontoparietal activation is too weak, the default mode network activates and dominates the system. This surge in default mode network activity correlates with the generation of task-unrelated thoughts. Behaviorally, this internal noise results in erratic task execution and frequent observable lapses in attention. Individuals with high working memory avoid this cascade. Their LCNE system successfully engages frontoparietal control, keeping the default mode network suppressed during the task. An attention lapse is a mechanical consequence of LCNE dysregulation failing to suppress the default mode network. General arousal dysregulation correlates with a lack of attention control. However, the data reveals a critical null finding. There is no direct correlation between arousal regulation and working memory capacity. Arousal regulation also lacks a direct correlation with overall fluid intelligence. Arousal and intelligence are connected through a specific mediating variable: task-unrelated thoughts. These thoughts branch outward to degrade attention, working memory, and fluid intelligence. Individuals reporting high volumes of task-unrelated thoughts exhibit negative correlations across all three cognitive metrics. This indirect pathway introduces internal noise, which degrades the active monitoring required to successfully execute fluid intelligence tasks. Arousal dysregulation places the brain in a state of chronic internal interference, which reduces available working memory capacity. On a microscopic scale, our executive functions rely entirely on internal currents and synaptic transmission within the frontal cortex. These functions depend on a strict division of labor between D1 and D2 dopamine receptors. This logic gate model shows D1 receptors controlling cognitive stability, dominating neural response during short-term memory retention to filter out environmental noise. When conditions become unstable, dominance shifts to D2 receptors, which control cognitive flexibility and update the internal state. Any change to a memory state requires the disengagement of attention, a process driven by D2 dominance. This microscopic receptor balance directly relates to the variations measured in standardized intelligence testing. Fluid intelligence relies on a constant tension. The D1 system locks down data, while the D2 system disengages that lock the instant the environment changes. This prefrontal efficiency is modulated at the genetic layer by the Val158 MET polymorphism in the COMT gene. The COMT gene regulates catecholinergic signaling directly within the prefrontal cortex. During high fluid intelligence tasks, we see increased bold response efficiency in the lateral prefrontal cortex and the interparietal sulcus, directly correlated with a higher VAL allele load. This genetic advantage is uniform across neural pathways, improving processing efficiency in both spatial and verbal domains. Pharmacological data in primates shows the fragility of this chemical balance. Blocking muscarinic receptors in the primate dorsal lateral prefrontal cortex causes the neural architecture to degrade. This blockade reduces overall neuronal firing rates within the region. Without baseline firing, task-selective cells lose their ability to discriminate rules, creating severe deficits in working memory representation. The physical representation of a cognitive rule exists only through precise genetic and molecular catecholamine regulation. Working memory capacity operates as an active state of constant network suppression. The locus coeruleus norepinephrine system regulates whether the brain can silence the default mode network. Simultaneously, D1 and D2 dopamine receptors in the frontal cortex must toggle between focus and disengagement. High fluid intelligence is marked by this synthesized state, a low activation bold curve supported by high LC and E tone, robust frontoparietal control, and precise dopaminergic switching. This physiological harmony helps filter out the indirect pathway of mind wandering, maintaining a task-positive response with fewer task-unrelated thoughts. The variations in human intelligence are rooted in this microscopic physiological precision. This returns us to the concept of neural efficiency. High fluid intelligence is a state of extreme biological precision, achieving maximum cognitive output with minimal metabolic waste by filtering out internal interference.